Welcome. On behalf of Bio, I'd like to welcome you to the panel, Disruptive Thinking, New Tools to Transform Addiction Treatment. My name is Kurt Rasmussen. I'm the Director of the Division of Therapeutics and Medical Consequences at the National Institute of Drug Abuse. And I'm delighted to be your host and moderator for today's discussion. Addiction, better characterized as substance use disorder, has grave effects on both the public health and the well-being of our society. In addition to having horrible consequences for the individual suffering from the disorder, substance use disorders affect the entire family and can rip them apart emotionally and financially. Today, in our modern society, too often addiction is considered a character flaw, and patients suffering from these chronically recurring diseases are treated as lepers. I have no doubt that future generations will look back with derision at how we treated patients. Novel treatments are desperately needed for all types of substance use disorders. The Division of Therapeutics and Medical Consequences at NIDA is focused on delivering new pharmacotherapies, behavioral therapies, and devices for the treatment of substance use disorders to the clinicians and patients battling this devastating disease. I am proud to be part of NIDA's mission. It's time for novel thinking and proactive action to address the problem of substance use disorders. Deeds, not words, are needed to deliver the novel treatments for substance use disorders. Today's panel includes four distinguished guests who are striving to transform the treatment of substance use disorders. I'd like to introduce each panelist, and each one will spend a few minutes describing their efforts after I introduce them all. Today's panel includes Dr. Anna Lemke, who's an associate professor of psychiatry and behavioral sciences at Stanford University Medical Center. Anna is the program director of the Stanford Addiction Medical Fellowship and chief of the Stanford Addiction Medicine Dual Diagnosis Clinic. She's a diplomat of the American Board of Psychiatry and Neurology and a diplomat of the American Board of Addiction Medicine. She's also author of the best-selling book, Drug Dealer MD, How Doctors Were Duped, Patients Got Hooked, and Why It's Hard to Stop. We also have with us Dr. James Hickman, founder of and chief scientist at Hesperos Incorporated. He is also the founding director of the Nanoscience Technology Center at the University of Central Florida. Previously, Jay was the Hunter Endowed Chair of Bioengineering at Clemson University. We also have Dr. David Gustafson. Dave is an emeritus research professor at the University of Wisconsin-Madison Center for Health Enhancement Systems Studies. Dave is a member of the National Academy of Engineering and a fellow of the American for Health Services Research, the American Medical Informatics Association, W.K. Kellogg Foundation, and the Institute for Healthcare Improvement. And we have Dr. Stephen Hurst. Steve is a co-founder, executive chairman, and co-CEO of Mind Medicine. Steve has more than 35 years experience in the biopharmaceutical industry, including work for the Immune Tolerance Institute, the Regents of the University of California, the World Bank, and BioVentures for Global Health. Now I'd like to take a few minutes to have each panelist describe their work in a little more detail. And Anna, we'd like you to go first, please.
Hello, everybody. I'm delighted to be here. I'm going to mainly talk today about my role as chief of our addiction medicine dual diagnosis clinic. Addiction is the compulsive use of substances despite harm to self and/or others. And a dual diagnosis clinic means that we treat people with addiction who also have co-occurring psychiatric disorders. What I'd like to do very briefly is talk about some major epidemiologic trends that we're seeing in the field of addiction medicine, as well as the tools that I have found most useful in patient care. So first of all, major trends in addiction today. First of all, we're seeing more women presenting with addiction. It used to be the ratio was 5 to 1 to 2 to 1, men to women. Now we're seeing a ratio of more 1-to-1, especially among millennials. Secondly, we're seeing more older people presenting with addiction. So people, especially after retirement, finding themselves with more time, less structure, who have been able to engage in controlled substance use and finally get to a point where it's out of control. We're also seeing many more people presenting with low-level addictions who are coming in wanting to help managing their consumption. So they don't want to abstain from their substance, but they want help in seeking moderation. And then finally, we are seeing many more what we call process or behavioral addictions: gambling, the internet, video games, pornography. So those are some of the major epidemiologic trends. Now I want to talk briefly about some tools that I find especially useful in clinical care, and I wish I had more of. So that's a challenge for all of you. What I wish I had more of is medications that helped with appetitive control. So medications, for example, like naltrexone, an opioid receptor blocker, that not only helps people abstain from alcohol and abstain from opioids, but also helps people who want to drink in moderation. When naltrexone works in my patients, what they'll say to me is, wow, when I take naltrexone, I can have just one drink. I don't have to drink the whole bottle of wine or whatever it is. So more medications like that help not just with abstinence, but also with moderation and with appetitive control. The other thing that would be incredibly valuable is more remote monitoring devices to gather objective data. So what I especially need, I have the patient's subjective account, I have collateral information from family members, but I would love more ways to get objective data on their physiology. For example, did they use substances? I often in my clinical work will use a remote alcohol monitoring device, a scheduled breathalyzer with a facial recognition software. And I use it really because patients want it. They say to me, you know, I'm now not using and my wife doesn't believe me. If I had a device that could hold me not just accountable to myself, but also prove to her that I'm not using, that would be really useful. So devices like that would be great. Also, especially since the pandemic, overnight, we've translated to Zoom care. So all of my work is now remote. I would love to have devices or ways of objectively monitoring not only whether or not a patient has used a substance, but also levels. So for example, with cannabis, it sticks around for so long, urine drug screens end up being positive for weeks or even months in some cases. I'd love to have a way to see that the levels are actually going down. So when the patient tells me that they're not using anymore, I can verify that with some sort of objective level. Also, as we use more and more addiction pharmacotherapy for our patients, I would love to know more about what are the long-term consequences of those kinds of drugs. I need to know about more drug-drug interactions with the drugs that I prescribe for addiction and the many other drugs that our patients are often taking, from psychotropics to other drugs to treat medical illness. So that's my challenge to all of you, and I really appreciate the work you do. It really helps me in the work that I do.
Anna, thank you very much. That was very, very interesting, very intriguing. We'll look forward to getting back to some questions, but I want to go around and give everybody a chance to say something. So next we're going to have Jay go, and Jay's got some slides. Let me bring those up and share them. There we go.
Thanks, Kurt, for the introduction, and Anna for highlighting kind of some of the things that we do. I wanted to have some slides just simply because it's really hard for people sometimes to wrap their head around the whole idea of a human on a chip. Really, if you kind of start in the upper left, your human body, and then normally to try and figure out how to dose somebody in a phase 1 clinical trial, you can play what is called a PBPK model in silico, basically in a computer program, to simulate all the different interactions and all the different organs to be able to predict what would be a safe dosing. So my collaborator, Michael Schuler, said, you know, I can take all these equations and stuff and then use it to design a chip. So if you then look in the right-hand side, you can actually see like these are actually chips that we can actually build. And we were actually recently chosen by NIH and the HEAL initiative to actually build a overdose model. So if you go down to the bottom right-hand corner, What we want to try and do is be able to look at efficacy and off-target tox in the same system for an overdose model where we can actually take an overdose on a narcotic in a human-on-a-chip based system and then recover it with naloxone, okay, and be able to then understand how to do that. What's really interesting is the system works at, if you're familiar with this technology, there's a lot of pumps and tubing and everything. What we do is we use a pumpless platform. We just use gravity to pump it. This allows us to not only look at a medication, but its metabolites, so we can have a competent liver in the system. And since it's low volume, we can actually look at concentrates of metabolites and see what their effects are in the same system. Next slide, Kurt. So again, this is the same system that we're building for NIH. Now, the other thing that's very interesting is generally people want to look at biomarkers in terms of preclinical types of assays. When we said, well, we would really like to be able to try to reproduce some of the functions a doctor would look at when he's examining you or in a clinical trial, like, you know, when you walk in a doctor's office, he doesn't immediately take your blood to be able to look at your biomarkers. He says, well, how are you moving? Is your muscle function okay? Or your eye movement? Are you cognitively interacting with him well? He then listens to your heart. Is the heart beating properly? You know, and then he will maybe order some biomarkers to look at, you know, liver and kidney function and things like that. Well, what we've done is we've been able to create these, you know, functional markers where we can actually monitor cardiac force or output. We can monitor cardiac conduction velocity. We can monitor skeletal muscle force. We can look at neuronal communication. And we're going to use this to be able to look at efficacy, okay, for neurons in the system, is to see whether or not, one, we can put them into overdose and whether we can recover it, but also at the same time look at off-target toxicity for naloxone in terms of repeat dose, because now in addiction you have people who are being exposed to it multiple times. We don't know what the long-term effects of that are. So we can also be able to look at comorbidities in these systems as well, because we can put in, say, a diseased heart in the system and look and see, okay, what happens when you overdose somebody on opioids and then try to recover it and you actually have a heart condition. As Adam mentioned, older people are now actually exhibiting with addiction trends as well. Next slide, Kurt. So the neurons, it's a very busy slide, but just very quickly, on the left-hand side, it kind of shows a picture of your brain stem. There's a small area in your brain stem called the pre-BOT-C neurons. And you don't have to know exactly what they are, other than what they do is they control the rhythmic breathing, okay, of your lungs. And this is the main target that causes people to go into overdose, okay? And so when these neurons stop firing, and you can see in the second panel on the left-hand side, when you treat it, okay, with a mimic of an opioid, Damgo, it basically stops this rhythmic function. And you can then actually try and then recover that by trying to displace the opioid with naloxone, and that's how that works. So on the right side, we're going to be able to take these stem cells, we're going to create the pre-BOT-C neurons, we're going to put them on the electrodes like I showed you. And the bottom, you can actually will have baseline recordings, hit it with the opioids, it'll go into overdose, and then try to hit it with naloxone and then recover it. Now the interesting thing is we can not only use pre-BOT-C neurons, but we can also put in nociceptors, which are pain receptors, to be able to look at pain receptors in our model. We can also put in the addiction circuit from the brain in our model, because they're interchangeable. So the neuronal circuits don't have to just be looking at these pre-BOT-C neurons. It can look at any kinds of neurons that are involved in the addiction cycle. And so this becomes very, very important. Again, as Anna mentioned, we can also look at drug-drug interactions. Say if you're an older individual and you're taking 3 or 4 different medications, or you have another condition, and we're going to then hit it with naloxone, what kind of drug-drug interactions do you have? Last slide, Kurt. And then finally, very important as well, we want to be able to create these in silico models of these in vitro systems to be able to inform doctors and clinicians and clinical trials, okay, from our PK/PD models or in silico models of our chip-based systems, and to be able to feed that into the in vivo models, or the models of human beings, to be able to have it predict what all these effects will actually be doing in a person, okay? And so that's kind of our technology that we've have at Hesperos that we are now have available as a service to people. So I'm finished, Kurt.
Great. Thanks very much, Jay. Very interesting. Appreciate your thoughts. Dave Gustafson, can you tell us a little bit about what you've been working on, please?
Sure. Our focus has been on developing technologies, specifically things like computers and smartphones to help people cope with addiction issues. And when I say people, I mean both the patient and their family. And so I'm going to take the example of a smartphone kind of approach to this issue and talk about it. So we have a system, we call it HS, and don't worry about what that stands for. On the smartphone, what you would find are four different kinds of things that we're attempting to do. The first thing is that we're trying to create a way in which people can talk with each other about issues that they're struggling with. So, you know, a person who is dealing with an addiction would be able to anonymously communicate with other people. The second thing that we're trying to do is to improve a person's ability to cope with the stresses of, for instance, the cravings that they may receive as they're trying to get off a particular drug. And there, well, we have mindfulness programs and relaxation programs and various kinds of interventions that will allow them to deal with their cravings better than they have up to this point in time. The third thing that we try to do is to improve and retain their intrinsic motivation to stay clean or to stay sober. And so we'll have all sorts of different kind of things, all the way from reminders of why they want to stay clean. We use GPS to monitor the movements of a person who, for instance, has an alcohol problem. We enter the data on the bars that the person used to frequent. And if a person comes close to a bar, then the system will monitor them. If they stay there for more than 5 minutes, they'll get a phone call. If they get that phone call, what they'll hear is, for instance, a recording from their daughter saying, "Daddy, please don't do that, come home." They define, the drinker defines what kind of an intervention they want to have at that 5-minute period. But that's an example of one of the real ones that we had. The fourth kind of thing that we try to do is to monitor people over time to see how they're doing. And that information, you know, collects about 10 different kinds of pieces of information, ranging from the extent to which a person is feeling under pressure from family relationships, or feeling that they're struggling too much with depression, or not attending their 12-step meetings. We collect 10 pieces of data on a weekly basis that will allow us to track how things are going. And then we use what's called ecological momentary assessment. And several times during a day, the phone would call the person and say, where are you at? What are you doing? How are you feeling? Who are you with? And that kind of information is also used in a variety of ways, using machine learning as a way of trying to help people figure out if they're approaching a time when they're going to relapse, and then we initiate different kinds of interventions. The system has been tested in a number of different ways. There's been some randomized trials taking a look at what effect it has on heavy drinking, for instance, use of treatment services and things like that. We've done those tests in Appalachia, where we compared 200 women that were struggling with addiction, half of them getting HS and having half of them not. Found that the number of services that people continue to receive almost doubles when you get HS. The company has been a company has been created to disseminate the system, and we're now serving probably well over 10,000 people with the system. The future directions that we're going with this is to move away from smartphones and move toward smart speakers and smart displays, especially for elderly people, so that they don't need to get up and go to a computer. They can just say, step back and say, HS, it's time, let's take my weekly survey. And everything is done without them having to touch a keyboard. There are new services that we're adding in and to make the system more effective, using, for instance, natural language processing to identify people who are really struggling. For instance, one of the things that we've found is that the more people swear, not baseline, but the change in the amount of swearing is explains about 40% of the variance in whether a person's going to relapse or not. And so we'll look at words that patients are using to help us determine what's happening. I think that's enough for right now. Thanks, Kurt.
Wow, Dave, thanks so much. Fascinating. It sounds like some really exciting work. Thanks so much. Steve, you want to give us a little bit of a background and some overview as to what you've been working on?
Yeah, thank you, Kurt, and thanks everyone. This is a great panel. I'm enjoying the discussion, learning a lot. So we're a neuropharmaceutical company, and I've been doing drug development literally since I was 18 years old, and I got involved in my first project as an undergraduate at UC Berkeley. And I've worked on drugs in multiple areas: cardiovascular, diabetes, cancer, even gout in my career, and have a number of drugs that I've been involved with that are approved and on the market. And I'm very proud of that. It's a lot of work to get a drug onto the market, and for some reason I keep doing it. I haven't decided quite why, but it does challenge me. So I wasn't feeling challenged enough about 10 years ago when I decided that I wanted to get into neuroscience, and in particular focus on substance use disorders, or what are commonly known as addictions. And we started with a conversation with our founding scientist, Dr. Stanley Glick, who was head of neuropharmacology at Albany Medical College. And he had been working on a drug that had been essentially engineered based on a hallucinogenic compound called ibogaine, which is extracted from the bark of a shrub that grows in West Africa. And his interest in ibogaine began in the early 90s, when a gentleman who was advocating that ibogaine was the cure-all for addiction came to his laboratory and said, I need to get some science to back up what it is that I know to be true about ibogaine. And Dr. Glick did some examinations in his laboratory of the effects of ibogaine in what are called self-administration models. These are rats or almost any mammal you can use to train in a drug addictive substance self-administration model. And what he found that interested him about ibogaine was not that it reduced the amount of self-administration in the animal models, but that for days after a single administration of ibogaine, the animals continued to stay well below their baseline levels of self-administration prior to receiving ibogaine. So this intrigued him. This durable effect he found very intriguing. And I start the story here because it's really the start of my journey. In parallel, I've always been fascinated with the way the brain works, understanding why it is that I can leave the house in the morning with my dry cleaning in the back seat, intending to drop it off at the cleaners, only to find myself at my office with the dry cleaning still sitting in the back seat of the car. And I've always wondered why it is that I can go into Starbucks fully intending to get a banana and still walk out with a scone. These are things that fascinate me because I think I'm a reasonably intelligent person and I should be able to not forget to drop off my dry cleaning and resist the scone at Starbucks. Well, it turns out that because of neuroplasticity, which most people have heard of at this point, and we've learned a lot about in the last couple of decades, that our brains engage in really multiple types of thought processes. Some that are very conscious, burn lots of glucose in the brain, require a lot of focus, problem solving, new situations, that unfamiliar situational awareness. You become alert to potential danger. You start getting into a lot of very active, very intense, if you will, thought processes. And then there's a part of the brain that's driven by what we would call learned response or cognitive bias. And this is the example of the scone, probably, well, less the scone, but more the dry cleaning. The reason why I didn't drop off the dry cleaning is my slow thinking, processing, problem solving part of the brain got focused on something while I was driving to work, and my brain remembered how to get to work and took me to work rather than taking me to the dry cleaner, which required a diversion from the normal pattern that the brain engages in. So these are things that have always intrigued me. And then when you get into addiction and you start looking at what's going on in the brain of a patient that has a substance use disorder, the etiology of the disease became of great interest to me in my work with Dr. Glick. So what Dr. Glick did is he started working with a medicinal chemist. They started playing around with the ibogaine molecule, making a bunch of changes in it in the laboratory, and they created what we call a library of analog compounds. And they create about 60 of these designer compounds, and the 15th one that they tested in terms of anti-addictive properties was our lead product right now that's in clinical trials for substance use disorders. It's in early clinical trials, and we're expecting the first patient clinical trials to begin late this year in opioid use disorder initially. And then we will expand into other addictions. Now, why can we do that? The cause of addiction in the brain is a singular, in many ways, primary etiology of dysregulation of dopamine in the reward pleasure centers of the midbrain. And almost, as far as I know, and I'm only cautious because Anna and other experts that are more expert than me are on this panel, the dysregulation of dopamine in the reward pleasure centers of the brain is driving the craving and the changes in associated with neuroplastic phenomena in the brain that create a dysregulation of dopamine, where now your dopamine is driving your desire for whatever the pleasurable substance is that you've been repeatedly using over some period of time. And eventually this dysregulation becomes something that does not reset itself, and the patient at that point needs help. But the interesting thing is, it doesn't matter how a patient gets to this dopamine dysregulation, doesn't matter the substance or the behavior. This is a mechanism in the brain that is common to all types of addictive and repetitive behaviors. So we have data with our lead compound, not just in opioids, but also nicotine, alcohol, in stimulants like cocaine and methamphetamine. We also have data in compulsive eating models associated with what we call the fat rat, whether they're on a high fat diet or a high sucrose diet, that when they get exposed to our drug, they make far better choices and do not seek the high sucrose, high fat substances as much as a normal diet. So we know there's this common portal that the disease travels through. And by focusing our drug development on that common portal and making an effort to reset dopamine regulation in the brain to normal, it enables us to take a new approach to treating substance use disorders. The primary medicine approaches today, and I mean by that drug therapies, are substitution therapies for the most part. where we're essentially taking a substance that is of less health liability and substituting it for something that has a very high health liability. And the best example would be methadone being a replacement for heroin. That's probably the best known example. A nicotine patch as a replacement for tobacco. So our whole program is based on really a very systems approach in what we describe as the discovery, development, and deployment of psychedelic-inspired medicines to alleviate suffering and promote wellness. And we use psychedelic-inspired because of our origins around ibogaine, and although our compound was engineered in a way that eliminates the hallucinogenicity while retaining the anti-addictive properties, we're also looking at psychedelic compounds that have anecdotal data, meaning stories of them being effective in helping someone overcome an addiction. We're also involved in programs around some psychedelic compounds, like, for example, psilocybin, which is the active hallucinogen in magic mushrooms. And there's a very good clinical trial ongoing at New York University on psilocybin in alcohol use disorder that's extremely promising. So our company is looking at the entire field of medications that are either derived from or are traditional psychedelic compounds. And we're not just applying those to addiction, we're also broadening that out into mental health in general. And as we learn more about the etiology of mental illness, similar to what we know about addiction today in terms of the etiology and what exactly is going on with the disease process in the brain, I think we'll get better and better at creating new medicines that should be an improvement over the current standard of care. And as I think Anna will tell you, there's a lot of room for improvement today. So that's what we do.
Fantastic. Steve, thanks very much. So thank you. That's very exciting introduction. We've got some really out of the box thinking, some exciting things coming along, everything from some new in vitro type ways of studying the issue and developing new products to more a device type approach with Dave and Steve, some new pharmacotherapies. Very exciting. So Anna, I'd like to turn to you first, if you don't mind. So you just heard some of the new developments. As someone who's in the trenches as an addiction psychiatrist, I'd like to get your reaction to some of those things and whether or not you think the advantages and disadvantages of each. One thing in particular that struck me was Dave's approach to improve motivation. That seemed, as a psychiatrist, what's your reaction to that approach, and do you have any suggestions or things to help or that could help Dave in any way? Yeah,
great. So yeah, I found the presentations fascinating and somewhat counterintuitive, and I'll elaborate on that. But I guess when David was talking about HS, this smartphone mediated kind of system to help patients, what really occurred to me is, they've digitized mindfulness. So, mindfulness is kind of this buzzword that we use, and a lot of people are like, what does that really mean? Mindfulness is kind of a way of becoming aware of your thoughts and emotions in real time without judgment. And that without judgment piece is really important. So it's a way of being in the world where you learn, and it's actually, you can learn it, it's a skill, it takes practice. You learn to observe what you're feeling in the moment, you try not to judge feeling that, and you just become curious about the inner workings of your world, of your brain. And what struck me about David's presentation was, that's fascinating. They've basically come up with like a software that kind of teaches people mindfulness. There are like these prompts, to kind of get people to stop and think about cravings, or to stop and think about, their other emotions, or to help them connect. In general, my bias about these kinds of apps is that people really want connection to other real people that they know. And so they want anonymity, but they don't want anonymity to the point that they're not seen and that they don't see the other. So when he talked about, connecting to some anonymous other person, I guess I felt that was less strong, because really people want to connect and be seen and be known and have these intimate connections, but they want to make sure that it's a private, safe space to share. And I'm sure that there are ways that HS does that too. So those are my thoughts about that. As far as Stephen's talk about, Ibogaine, that was fascinating because the way that I've read about and seen psychedelics used to treat addiction is in psychotherapy assisted manner. So basically you get a pulse dosing of 1 to 3 doses of psychedelics, mushrooms, psilocybin, ibogaine, interspersed with this intensive psychotherapy. And the way that I look at that is it's like taking the gondola to the top of the mountain instead of having to hike up. It's like a condensed version of the psychotherapy that's sped up by the psychedelic. But if I understood you correctly, Stephen, it sounds like what you were saying is that you're actually going to use this ibogaine derivative as a daily pharmacotherapy, the way that we use buprenorphine or methadone as daily pharmacotherapy for opioid use disorder. So I don't know if I got that right, but I'd be intrigued to hear more. And then as far as what Jay was talking about, slightly over my head, but from what I could understand, this in vitro tissue, what I kept thinking, and again, this is sort of my, tangential thinking, was, wow, wouldn't it be cool if you could take that thing that they're doing in vitro and actually put it in vivo, including having emergency naloxone release in case of an overdose. So that's where my mind went, hopefully not too tangential, but I'd love to hear more from the panel.
Great, thanks very much, Anna. That's fantastic. Let me ask a quick follow-up question. You did mention the intimate space and you're worried that maybe some of what Dave was working on would be too impersonal. But one thing you did mention, and that caught my ear, was that to play the recording of a daughter or someone, a close person, saying something at a particular moment, might that be a way to address it? And Dave, do you think that that's a way to introduce that intimacy that's important?
It's Anna, do you want to go or do you want me to go?
Oh, no, I thought that was great. I mean, there was so much that I liked about it. Don't get the wrong impression. It's not that I love that I thought, well, that's brilliant. To me, that's the kind of mindfulness. It's like that conscience or that awareness. What are my values? What are my goals? And the apps or the systems, serves as sort of a prompt to remind us to be in that space of awareness. So I thought that was also beautiful and brilliant.
Well, thank you. To give credit where credit is due, Richie Davidson from the Center for Healthy Minds is the person who has created that part of the HS system. And I'm blown away. I just think that he's an amazing guy. As far as the discussion groups are concerned, I would say 80% of the use of HS is in the discussion groups. And when we look at it, we learn so much about interactive behavior. For instance, one of the things that we've found is that for people who have a lot of social support already, HS doesn't do much. But if you take a person that is really, lonely and isolated and this kind of stuff, it makes a humongous difference. And so I understand the face-to-face part of things. We've tried, for instance, Zoom and those kinds of opportunities to see each other. And I think for a lot of counseling and that kind of stuff, boy, it makes a big difference. But we've heard from most, I would say the majority of our people don't want to be seen face to face.
Interesting.
And so that's another thing. And so, well, I'll stop there because there are a bunch of other people on the stage here.
Thanks, Dave. Thanks very much. Steve, I was wondering, Steve and Jay, Jay, you've got something that could be looking at repurposed drugs in a way and analyzing things in a different way. And Steve was working on something, a derivative of ibogaine. Do you think that your system would be amenable to studying types of medications that Steve is bringing forward? Oh, you're on mute. You're on mute. You're on mute.
So we've actually ran 6 workshops with the NIH and the FDA to look at how to use these systems. And one of the things is exactly like what Steve has, is if you started a clinical trial, you've got some safety data, you can actually look at a different application for a drug and potentially go directly into patients. If you can show there's already existing toxicity data, okay? And so if you have an efficacy model that's a human-on-a-chip based system and the FDA believes the data, they will take that directly into patients. You know, so if there's already, again, existing drugs and you have that safety data, you can do that. And they said there were, you know, the example's already set up in terms of Vertex has a drug for cystic fibrosis where they had initially approved for, I think, 12 mutations. And FDA just recently approved them to go directly into patients with an additional 32 based upon only the efficacy data in vitro. And so what we're trying to do is to build models. So Anna, kind of what it'll be used for is there's all kinds of experiments you would like to do on a human, but you can't. And it doesn't really work that well with animals because they just don't think like us. So what we're trying to do is to give you that in between where we can set up those neuronal circuits to be able to do the experiments, to be able to look at the efficacy that Stephen would want to do, but also at the same time look at the off-target toxicity of those drugs. And also drug-drug interactions. If you're already taking some, say you're already taking a couple anticholinergics and you want to take Stephen's drug, what happens? Okay, and have that information before you go into patients. And so those are the things we're trying to do, is to come up with better information before you go into patients and then potentially skip clinical trials in some instances with our systems.
Great. Thanks very much. Steve, I want to give you a chance to jump in here. A couple of comments, have any reaction to what's been said, but also the safety has come up a couple of times, and I know there are special concerns with, well, you've gone beyond Ibogaine, but Ibogaine and psychedelics, there's always potential some safety issues. What's your response to those type of questions?
Well, let me answer Anna first, and then I'll come back to the risks.
Okay.
Okay, if that's okay. So,
of course.
Anna, I think you did capture pretty much the essence of what we're doing. So, for example, as we move into our opioid use disorder studies, we're starting with medication-assisted treatment that is already standard of care, both in withdrawal and in maintenance. So what we're looking at is, can we improve current outcomes by adding 18-MC into the mix? Now, the advantage that 18-MC has over Ibogaine is it's not hallucinogenic, and there's none that we've seen, cardiovascular toxicity. And as you know, with Ibogaine, QTC prolongation is a huge issue in terms of its proarrhythmic properties, and is probably responsible for some deaths associated with administration of Ibogaine at high doses. We engineered all that out, and I think the brilliance of Stan Glick was an observation in early 1990. We're asking the question, the hypothesis that, you know, is the hallucinogenic experience essential to the therapeutic value of Ibogaine? And the answer that we get is no, it's not. Similarly, if you look at LSD in cluster headache syndrome, which I'm sure you've seen a few cluster headache patients in your career, you know it's an awful disease. The patient medication surveys that were conducted through Yale University a couple of years ago revealed that it looked at prescription and illicit treatments that patients were using. And both LSD and psilocybin were provided the most relief, both in terms of halting a cluster headache attack and in relieving the frequency. In some patients, single doses resulting in no further attacks, which is pretty remarkable if you think about that. It raises a lot of scientific questions, but it's pretty remarkable. But the non-hallucinogenic analog, brominated LSD, BOL148, which you've probably heard of, is as effective as LSD in a hallucinogenic dose with respect to a small cohort of patients that was treated in Germany, reported in 2011. So that's kind of the basis for a lot of what we do, is we're interested in not just what you described, which is the hard reboot, we call it, a potent experiential dose in combination with cognitive behavioral therapy and a rather extensive amount of challenging work with a therapist that is potentially accelerated by suspending cognitive bias by using a hallucinogen that's an approach to therapy, and it's one that we think will have value. But we also look at the opposite end of the spectrum, which is these non-hallucinogenic analogs that appear to have retained much of the medicinal properties that have been associated anecdotally with psychedelic compounds. And our business, obviously, since we're a drug company, is actually data. It's anecdotal, accounts are great, but they don't get drugs approved. You have to do the full development program, full tox programs, very expensive programs in order to do that. So what we're looking at, first of all, is our lead compound in the withdrawal setting with a primary endpoint of induction of Vivitrol prior to discharge from the detox center. And then we'll be running a study on the use of our lead compound in combination with Vivitrol to see if we can have an impact on the recidivism rate within patients that have received at least a single dose of Vivitrol. So we're hoping to be able to augment your pharmacopoeia in a way that really affects patient outcomes, and as I like to say, moves the needle. It's desperately needed. We all know it's desperately needed, and hopefully by reversing this disease process in the brain, by restoring normal dopamine regulation in the midbrain, which is what our drug appears to do, that will have a great effect, particularly on recidivism, which is driven largely by what we call, as you know, cue-induced reinstatement. Clinking ice cubes to the alcoholic, a mirror and a razor blade to the cocaine user, a syringe and a needle to the heroin addict. These types of environmental cues trigger the brain disease to induce craving that drive people to go back to using when they've been in recovery. So that's our goal. Let's talk about risks. I've been doing, as I said, drug development my entire career. I actually view, from the data that I've seen, hallucinogenic compounds as being no more, and in many cases less risky than your average new chemical entity that is in development as a potential therapeutic. There's a lot of bias. You talked about that, Kurt, about bias around addiction. There's a lot of bias around psychedelics. It seems that the end of my career will be associated with fighting bias, both in the scientific communities and in the public. I had a scientific journalist the other day ask me why it was that we had this addiction program, but at the same time, we were developing psychedelics that are addictive. And I asked the question simply, what's the data that's telling you that LSD is an addictive compound? And quite honestly, there isn't any. That's a belief and a bias, but it's not driven by a lot of hard science. So our job is get the hard science, get the data. And I think as the data comes through and we go through regulatory agencies like FDA, it's my belief that we will find that these compounds and their therapeutic value far exceeds the potential safety risk to the patient. But again, you have to be wise about what you're doing. I do not think ibogaine is a good candidate because of its cardiovascular toxicity. We know that it can induce, that it's proarrhythmic. Developing compounds that are proarrhythmic puts you at risk of wasting an awful lot of money, because when you get to the ultimate review at the agency, they're going to look at it and say, your therapeutic index is not broad enough, this drug is not safe enough, we're not going to approve it. That's what we do.
Thanks, Steve. Thanks very much. That's great. I'd like to return to Anna for a second. So you mentioned something that you thought if you could get more help with appetitive control with your patients, that would be very useful. I guess I've got two things and questions. Do you think that any of the approaches we heard about today would help address that? And also, what other symptoms or what other problems would you like help with as an addiction psychiatrist besides appetitive control?
Well, you know, as Stephen talked about, what we believe happens in the brain of people who become addicted is that they essentially get into a dopamine deficit state, or like a dopamine dysregulation. And that part of the challenge, especially in early recovery, when they first stop, is kind of renormalizing a new homeostasis and revitalizing their own dopamine transmission. So, you know, I think Stephen's drug, I don't know how it works, but he seemed to imply that it does this. It kind of, you know, re-equilibrates baseline dopamine homeostasis, which is very useful. And we have other drugs that do that, but we don't have enough. And of course, there's enormous inter-individual variability. What works for one person doesn't work for another. So, I mean, I think the field, I mean, what are there, like, what, 6 FDA approved medications for, or maybe 6 or 7, maybe 1, 2, 3, 4, 5, 6, maybe 8 for all addictions. I mean, clearly that's absurd when, you know, you consider that 40 million Americans struggle with some kind of substance use disorder. So we absolutely need more medications of different mechanisms, you know, to get at different pathways. So that's very useful. As I said before, you know, what my practice really struggles with is objective data. And anything that can give me more objective data points is just super helpful. You know, I can work to build a therapeutic alliance. I can, you know, have all the skills in the world to get people to open up to me and to break through denial. But at the end of the day, part of the disease of addiction is to lie about use or to misremember, or however you want to characterize that. So monitoring devices, objective, physical data that we can then use to kind of track the disease, you know, and the disease is a chronic relapsing and remitting disease. So we expect people will cycle through in the course of a lifetime episodes of use and, you know, addictive use, and then get back into recovery and have a period of recovery, and then, you know, have a cue-induced trigger, the clinking of the ice cubes in the airports that we don't go to anymore. Airports are terrible places for people with alcohol use disorder, and then they will relapse again. So, you know, something to track, a harbinger of relapse. And I know a lot of people are working on harbingers of relapse, but it's things like, oh, you know, when you look on an MRI, fMRI, like the brain's like, we're not going to be putting people through fMRIs, you know, in order to be able to see whether or not, so that's not practically useful. So I need things that are practically useful for a post-COVID world where I'm talking to my patients the way I'm talking to you right now, and they're in their home, you know, 60 miles away, and I'm trying to figure out like what's really going on with them.
That's great. Yeah. So as you mentioned that, I thought of something else that Dave said about measuring the number of curses someone was using as a predictor. Can you tell us?
That was fascinating. I mean, I'm a tiny bit skeptical of that and what it really means, but yeah, it's interesting. Like, what are these little social markers of you know, people's daily habits that are linked to substance use and relapse, but that aren't obviously linked, and looking for more of those. And we talk about that clinically with patients, you know, what are your red flags? Because often when people relapse, they're actually gearing up for the relapse weeks before they take that first drink or relapse to the pill or whatever drug they're using, smoking. You know, Alcoholics Anonymous is an amazing organization that's helped millions of people get into recovery, sustained recovery. And they focus a lot on targets and triggers. There's a great acronym, HALT, which stands for hungry, angry, lonely, and tired. You know, those are triggers for relapse. So again, a lot of it is also just mindful awareness and insight about your own kinds of issues that are triggers for you. But yeah, you know, anything that can help folks with that would be great.
Kurt, could I just make a couple of comments?
Sure, please.
Yeah, I think that one of the things that we underattend, 2 things 2 points. One of the things that we underattend is how important the family is in all of this. And as an example of that, we developed a program for lung cancer patients a while back, but it wasn't for the patient at all. It was for the family caregiver. and the system carried the family caregiver through the first 13 months after the death of the patient. So taught them how to be a better caregiver and that kind of stuff. But it helped them during the bereavement period. So it was never intended for a lung cancer patient. The lung cancer patients didn't look at the system. Only the caregiver looked at the system. And when we did a randomized trial, we found that length of life extended 40% for patients that were where the family had been a centerpiece of the intervention. Now, 40% isn't a lot in lung cancer. I mean, it's maybe a graduation, and maybe it's a birthday and this kind of stuff, because average length of time is about 9 months. But I think that this field is got to get away from looking at addiction as a person disease. It's not a person disease, it's a family disease. And until we really address that, we can be doing all sorts of interventions, and it's not gonna work. Not completely, at least. And then my second point is that during that lung cancer work, what we did was we collected these data from the caregiver of their opinion about how the patient was doing. And in another randomized trial, we gave that information to the clinicians. No, we didn't give it to them. We forced it on them. And so we would call a nurse, or we would call another designated person and say, you know, this person's relationship is really going to pot right now, and I'm very worried. And when we did that, we also found that patients' symptoms went down, and that the wellness of the patient improved. And so it's really important that we find ways to not alert, but to force information down the throats of clinicians, whether they have the time to receive it or not. Because when they finally receive it, they act. And they act in ways that allow people to recover more quickly. And so that's another thing we do, is to share that information with the clinical team.
All right, thanks very much, Dave. We're getting close to the end of the hour. I want to go around, and Jay, you haven't gotten a chance to get in as much. Maybe some closing thoughts or some closing comments. I'll start with you, Jay. Anything else you wanted to add?
Just in terms of, you know, it's a new tool that people aren't really that familiar with, in terms of it can be very helpful for drug development and speed up the process, be able to answer questions you can't do in animals right at the moment.
The human, you mean the HumanOnAChip?
Correct.
Yeah.
You know, and again, we're looking right now at overdose-based systems, but we can put pretty much any neuronal circuit in there, neuronal reward. We can look at the dopaminergic system. We can also look at
that's what I was going to ask you. We've heard a little bit about the dopamine system. Is that something?
Yeah, we can put that on the chip, and at the same time, we're evaluating whether or not that drug is working. We could be looking at the off-target tox, because we published a paper in Science Translational Medicine where we looked at therapeutic index, okay, for cancer drugs, okay, and then Francis Collins featured that in his blog. So it's starting to get people's attention. We can do therapeutic index in these HumanOnAChip based systems, and the more people know about it, the more people are trying to use it.
Great, thanks very much. Steve, any closing thoughts you want to add?
I just make the comment that, you know, every century, at least for the last few, we've had the hallmark of the century innovation or area of medical advance. And, you know, in the 1800s, it was probably anesthesia, the 1900s, antibiotics. I think this is going to really be the century of neuroscience. I think the advances, the rate at which we're learning, the things we're learning, you know, 20 years from now, I think Anna's right. People are going to look at the way we treated patients today and just think we were primitives. So I'm very excited about the work that we do now, and I'm very optimistic about the future. The only thing I can say is with COVID-19, it's clear that our work is more important than it has ever been before.
Right. Thanks very much, Steve. Dave, any closing thoughts you'd like to add?
Nope, thanks.
Okay. Anna, how about you? Let's end up with you. We've heard a lot about some really exciting new directions to go on. Any closing thoughts you'd like to add?
Well, I'm now wondering if we should give this ibogaine derivative to family members of people who are addicted.
I'm all in favor of that.
Right. But seriously, I actually want to echo what David said, because I think it's so important. And that's the point about family. And we try very hard in our work to bring family members in. And when they come in, we're so much more likely to have traction and have success and be able to help our patients. And yet, you know, there are a lot of people out there who don't have families and who are isolated. So we have to find a way to kind of create surrogate families for those people, because isolation is both the cause and the result of addiction. And those two things kind of go hand in hand. So I'm really excited about what I've heard and learned today. I think it's so exciting that there are so many brilliant people who are working on the problem of addiction. I've been in this field for 25 years, and for so long it was sort of like, you know, the bastion of these sort of wounded warriors, and there wasn't a lot of national focus or scientific energy being invested. So I'm just really thrilled that as I kind of get to the latter half of my career, that there's so much momentum and so much excitement and energy. It makes me very hopeful about the future.
Fantastic. Delighted to hear that. I agree. And as I said at the beginning, the way we treat people with substance use disorders now today, we treat it as a character flaw. We treat them as lepers, and people will look back with disdain at how we treat them today. And so I look forward to a future where Anna, the addiction psychiatrist, will be a heralded position that everyone wants to have. And Steve, there'll be some of your new pharmacotherapies. And Jay, your techniques are going to be working toward discovering new compounds and repurposed compounds. And Dave, your new products are going to be helping by themselves, but also be integrated with the new pharmacotherapies. Because I think the integration of device pharmacotherapies and psychosocial therapy will ultimately get us to where we want to go. So thank you so much, everybody. Great presentations. Thank you.
Thank you.
Thank you.
Thanks, Kurt,
Bye-bye.
for moderating.